Thanks for introducing. I'm Jiachen Li from Chinese Academy of Science. In this flash talk, I will very briefly introduce our recent work on improving RNA velocity modeling by using the transcriptional regulation information. RNA velocity aims at predicting the cell fates by modeling the splicing dynamics. Because of the inherent time delay between the unspliced mRNA and the spliced mRNA abundance, this model assumes that there should be a clockwise trajectory in the joint distribution between the unspliced and spliced, which is also called the phase trait. And in the phase trait, we can use different kind of ODE-based models to fit the point. Then we can get the RNA velocity and predict the cell transitions and cell fate. That's how RNA velocity models work. Although these methods have provided insights and been widely adopted in many clinical studies, but we found that actually, if we take a look at the phase trait fitting, we will find that these methods hard to fit the unspliced and spliced data well, which is because actually the splicing data distribution violates their assumptions. As shown in the right panel, here each point is a cell which is colored by the cell type. Because the unspliced mRNA count is highly sparse and noisy, there is significant overlap within cell types on the phase trait. So it's hard to extract the dynamics from the splicing data of a single gene. This will be a fundamental challenge for the RNA velocity methods. Then let's try something different. We plot the co-expression between some gene pairs and surprisingly find that the co-expression pattern between some TF-target pairs are very similar to what we desire to see in the RNA velocity models. As shown in the left figure, the difference is that here, in each plot, the y-axis is the TF, but in RNA velocity models, the y-axis is the unspliced abundance of that gene. So this inspired us to develop a model using the gene regulatory relationships to construct velocity model. And based on this idea, we developed our TFvelo model, which could integrate multiple TFs for modeling the target gene. And I will not spend time on the details in developing the model or those quantitative comparisons. I will only show two cases which could best support our idea. The first is the results on this data set. Our model can correctly predict the cell fate, but more important is that we can also fit the gene dynamics well on the phase trait, as shown in the far right panel. Actually, we first learn the representation from multiple TFs and fit the data based on the such basis, which could be much reliable and stable. This could be important for genes with high sparsity, as shown in the panel C. On some genes, because the unspliced abundance is quite sparse, the previous method based on that data cannot extract any patterns. But we can still use multiple TFs and learn the dynamics from it. And the next is the analysis of timescale. Actually, the splicing process is quite fast. Previous studies have shown that the splicing to be finished within 100 seconds. As a result, if we plot the unspliced abundance along with pseudo time, we could find that they almost change simultaneously. As a result, when we perform our model, we only have the phase trait, but in the phase trait, the cell types will mix and makes it hard to learn any dynamics from it because it's hidden by the data noise. On the contrary, the transcriptional regulation takes much longer time. So in this plot, we can clearly see that there is a time delay gap between the TF and target. So the cell types could be better separated in the phase trait and makes the dynamics modeling more easier. And this is a summary. In the previous slides, we have shown that using the transcriptional regulation could also model the gene dynamics and predict cell phase. But we also acknowledge that both regulation and splicing could provide supplementary information. So currently, we are working on a next model, which is a unified framework to connect both sides of them. In this case, we can model all the maybe 1,000 genes together and track the gene dynamics in a 3D faceplate, including both the regulation and the splicing process. And thanks for your attention. And if you have any questions, feel free to reach out to me.